I must say so. Thank you for the invitation, and I'm pleased to talk about the huge Meerfall project. So the American people actually don't think those are huge mares. So I have to convince them that these are Icelandic horses and not Icelandic ponies, and they do this all the time. And so it is a long-term project, and I will talk about that very soon. I will give you a little bit more introduction about allergy in general before I start with the project again. But Siba already mentioned that this was originally started, and that's really true, in 2007, when we had a workshop here, Havemeyer workshop. Havemeyer is a foundation in the US that supports international workshops on equine diseases, and one of those diseases is allergy, actually. So we had the third workshop here in Hola, actually, at Sister's Place in June 2007, and it never got dark, and we worked a lot, and we had a lot of fun, and I still refer to this as my favorite workshop, Havemeyer workshop that I ever had. It was just a fantastic event. But during that workshop, as I said, we also worked. So, and one key event to get this all going and to get this project started, which we are still working on, honestly, is that I met 3 key people there, and you may recognize them on that picture here. Let me get the pointer for a second. Oops, that was the wrong one. That's why I don't like them.
It's just the center.
The center.
Yeah.
So you may recognize them here, right? Those two, and then there's a guy hiding in the back here. And I was there too, and we were all much younger than today. But so that was very nice. So based on that, we incubated some ideas. I incubated some ideas about the Icelandic horse and how they can be used, how the Icelandic horse can be used really to study allergy in a little different way than we did before. And we had a couple of key observations. And then another very important thing here was that we thought about the Icelandic horse as a very important animal that is free of many infectious diseases. So in the US, people care a lot about infectious diseases in horses. They don't care that much, or not at all, about allergy, but about infectious diseases they care. And one of the infectious diseases they care about is herpes virus. So to bring this whole model in the US, we actually sold this as a model of EHV1-free horses. So this was already kind of a thinking, how can we make a project that we can fund, and also benefits my personal interest, which I always have since 1990 or something, on summer eczema of the horse, which started out in Hanover, as Siba described. So I will give you a little bit an overview of allergy in general, and about IgE, which is a key molecule in this type of allergy, IgE-mediated allergies in horses, which summer eczema belongs to, and about sensitization in horses, so what's that? And then a little bit about the maternal and foal parts, so sensitization and immunity in foals and young horses. And we'll then tell you a lot about the Icelandic Horse Project, and some of you have been here when the mares were here and the foals were here that were born at Kaldor, and so you may recognize some of those horses in those pictures. And then I will give you a little bit of some early results, and early sounds strange considering that this project started in 2010, but it's really a longitudinal project. We have to wait until the horses develop their phenotypes and their allergic phenotypes, and so just the first animals did that, and then we can really start to analyze our data. So I will give you a few of those, and then also talk a little bit about biomarkers and some markers that we try to find, and we don't have them yet, which are actually predictors of sensitization and early allergy. So which is, I think, a very important goal, and it's also an important goal, I think, for here, to identify horses probably early before they are exported, which may have a high risk to become allergic. So, so what is this all? I call this culicoides hypersensitivity because that's what we are really investigating. So it's an allergy that's mediated by midges of culicoides species, and it's characterized by itchiness, so the horses scratch themselves a lot because they are bitten by those midges. Very similar, if you get bitten by a midge, then it itches, right? So the horses have that too. High discomfort, they start to lose their hair, and they get skin irritations. And this is one of the mares from our project, that's Sigga. That shows her this year in April. She looks perfectly fine. She had already one allergic episode in last year in 2013. And then in July you can see these irritations around her face here, a lot around her face. That's where it starts, and it starts under the belly, and she rubs her mane and she rubs her tail. So many locations of her body affected. It occurs during the summer months when the midges are in the area. For us that's between mid of May till end of October usually. And then it's frequently described in Icelandic horses, so some people think about it as a disease of the Icelandic horse, which is entirely not true. So it occurs in all horse breeds. It's more dependent on how you keep your horses than dependent on which breed the horse is in. So it's found in most countries around the world, with a few exceptions, which are Iceland, New Zealand, Hawaii, and Antarctica, which doesn't have any horses at all, so no wonder. But all over the world where these midges occur, the disease also has been found with different prevalences. And then very early, when that was observed in the 80s, probably many people did skin tests on those horses, and really that pointed out that culicoides are the origin of that allergy. So bites by the midges. So what's going on here? So during the first time of exposure, and that's usually a long time of exposure, during the first summer, the midges bite the horses and they inject the allergens, and those are actually, we know that by now, allergens from the saliva of the midges into the skin, and that provokes an immune reaction which ends up with the development of IgE. So immunoglobulin E, that's a very specific antibody, an antibody that's involved in parasite responses in a good way, but it's also involved in allergic responses in a pathogenic way. So in those IgE antibodies, they go on mast cells, so sit on mast cells here on the surface, and they sit there, and this phase is called sensitization. So I will talk about sensitization quite a bit. This is an immunological quiet phase. So the sensitization itself does not mean any clinical disease. So that's a prerequisite for allergy, but it's not necessarily showing clinical signs at that stage. So for our horses, when they get exposed again, and that's usually in the second summer of exposure, so the midges bite the horses again in the summer, inject the allergens again, and then everything happens very quickly. So the mast cells get stimulated, and then they degranulate, and mast cells have a lot of inflammatory mediators, such as histamine and leukotrienes and prostaglandins, and they induce an inflammation in the skin wherever the midges bite the horses, and that causes these clinical signs, so itchiness, inflammation, and all the clinical signs that we see. So many years ago, by now I would say, yeah, so early in the 2000s, so we were kind of struggling a little bit with the skin reactions for allergies that really pointed towards the IgE-mediated reaction, but nobody ever really showed that summer eczema is an IgE-mediated disease. Everybody assumed and believed, and we thought it's probably true, but nobody showed it. So we thought about an experiment how we can show that, and we went back to an experiment that was done many years ago, by now almost 100 years ago, which is described as a Prausnitz-Küssner reaction. So that's a classical allergy experiment, even before IgE was found to be a mediator of that disease, for inducing allergy and transferring it to another person. So Prausnitz and Küssner are two Germans. I can say this in German here, luckily. So, and Küssner was allergic to cooked fish, so he had really a food allergy, right? And so he developed immediately systemic injection whenever, a reaction whenever he ate fish, so he couldn't do that. So what they they transferred some of his serum to Prausnitz and injected that in the skin, and then he ate, so he got afterwards a small extract from boiled fish, and he immediately showed a virulent flare reaction. So that's what you classically would see if you go to a doctor, at least in the US, many people are allergic. If you go to a doctor and you get a skin test done, so you see this virulent flare reaction of the skin. So we tried to do this in horses. So we asked, can the allergic reactions really be transferred to horses? So is this really an IgE-mediated disease? And can we induce it by culicoides extract? So and this is the result from that experiment. It was published in 2006. And so what we did here is, so these are many results, and this is a skin score after injecting those substances intradermally. And on day 1 here, so we added either IgG or IgE from horses that had or had not summer eczema, so no summer eczema, or had summer eczema, so different horses, into the skin of recipient horses. And the recipient horses are horse A and B, and on day 2, at the same location in the skin, so that was marked, all these horses got culicoides allergen, and they only responded at the sites where the IgE from the allergic horses were injected. So they didn't respond to the site where the IgE from a healthy horse was injected, and they almost, except for this 1 week reaction here, did not respond where the IgG was injected. So this experiment showed us that summer eczema is indeed an IgE-mediated disease, and it can be induced by culicoides very directly, very direct proof and evidence for that phenomenon. So we also did a few more experiments. For example, we added anti-IgE into the skin, and we did see the same reaction actually that we would see if we add histamine, or in an allergic horse if we add culicoides. So that further confirmed that IgE is really a key player in allergy, and that was not surprising, but was shown for the first time here. So we also, and you can see this here on the skin reaction, we also injected culicoides to these horses, and then we made a very interesting observation. And that observation was that many of our horses showed reactions to culicoides, even horses that were not allergic. And that has been observed, I believe, by many, many clinicians around the world that do these skin tests, that they can actually detect many horses that show a sensitization to that allergen, but they are not allergic. It's not your typical patient, but whenever somebody is doing a more research type of experiment, they see that. So they call this cross-reactivity, or they call this false positive, or whatever they call it, but is this really true? So we tested 24 of our horses at the university. Those are all clinically healthy horses. They had never shown allergy to that point when we tested them. They were in the herd for several years. There were animals in that herd that had allergy, so summer eczema, so we knew there is culicoides in the environment, but those horses did not have clinical signs. So there were a bunch of horses, different ages, median 11 years of age, and so we injected 3 allergens, quark extract, cat dander, and culicoides into the skin of those horses, plus the controls, which are saline and histamine. And as you can see here on the right side, so these nice reactions to culicoides were observed quite frequently in those horses. And overall, and I would like to point to this line only here and not explain any further on that slide, so you can see that we found 38% of those horses were actually sensitized to culicoides. These horses have still not developed allergy. So many horses that are not allergic are also sensitized to culicoides. So they do this first step, but they never move on to develop clinical signs of allergy. So there are obviously many, many things happening between that phase to get sensitized and between the phase after sensitization to develop allergy that determines if a horse will ever become allergic or not. So that points to the immune system, so very complicated, obviously. So we did the same thing again with young horses. When we found that we see these many sensitization reactions, we looked at young horses because at that time, not confirmed, but everybody talked about young horses don't get allergy, so we usually see that in older horses, young adult horses, that's when they develop that disease. So we tested 3 different age groups here, horses at 1, 2, and 3 years of age, relatively small n, but as you can see in this line here, so even in these young horses we could find horses that are sensitized to culicoides. So overall 14% of the horses at Cornell, non-allergic, not clinically allergic, were sensitized against culicoides. So that takes us to the next slide. So young horses don't get allergy. So generally it was described at that point when we started thinking about it that culicoides hypersensitivity develops by 2 to 4 years of age, and that's probably was rather gut feeling presented in certain papers by investigators than really measuring it, so it's more clinical experience. In addition, we did show, and I didn't show you the data for that, but we did show that IgE production can really not be detected in very young animals, in young foals, until they become 6 to 12 months of age. So there is no production by those foals during that time. And then these mast cell sensitization studies actually also showed that the young horses between 1 and 3 years of age then make IgE and can be sensitized. And then a more recent study, that's 2012, that also described that weanlings and younger horses, 2-year-old horses, can develop clinical signs of summer eczema. But I would like to point out that this was done in various environments and judged by various vets or owners. So that's really a little bit of a difficulty of those studies. So all that knowledge brought us to the question about sensitization of clinically healthy horses. And what is this really about sensitization and allergy? And so the sensitization of skin mast cells is a prerequisite for allergy. I mentioned that already. So you need to have that. Without sensitization, there is no allergy. However, it does occur in clinical healthy horses. So it's not a predictor necessarily, or we thought at that point it's not a predictor for allergy, which can be used to diagnose a horse previously to developing clinical signs. And then the late onset of allergy in the observed late onset in the horses, that we usually see this in the young adult horses or even older horses as clinical disease, that could also not be explained by young horses don't make IgE or don't get sensitized. They make IgE and they get sensitized, they still develop no allergy. So this overall, all these observations suggested that there must be additional mechanism beyond allergy and sensitization that determine that the immune system's decision, and I call this a decision, to become clinically sick, clinically allergic or not. So, and these are again 2 of our mares, and so this is an allergic mare. Here you can see that the missing hair, it's a little bit later in the summer, so she rubbed her mane off entirely and has many other clinical signs. While in the same environment, these horses have been kept in the same environment since 2010 actually, with the same exposure. That other mare doesn't show a thing. So he has the same exposure but no clinical signs. So what's behind that? So one important thing, if you consider allergy, is that allergies develop over a long period of time. So they become clinically typically during childhood and adolescence. That's for humans, and that's the same for horses. So you can see cases sometimes very early, but that's rare. So the typical development takes a little bit of time, and it's determined by several immune factors. So of course there is IgE, and the inducer of IgE initially is IL-4 and so-called Th2 responses. And that determines sensitization, so the step on the mast cell that I showed you initially. And then after the sensitization, so what we think is that this is kind of a circulus that's going on there with every single allergen exposure that really drives the development of IgE and becomes more and more and more and more until it reaches a certain threshold to develop clinical allergy. So but quite a timeline here between the initiation, sometime here, the sensitization and the clinical allergy. So it's not like an infectious disease. You get influenza today and then 3 days later you have a fever, right? So it's really a long period of time and many, many things happen here which you probably don't understand entirely yet. However, we do know a little bit, and that is that certain other immune responses like T regulatory cells, IL-10, TNF-beta, or certain IgG responses rather present or lead to tolerance against allergy. So especially there's some thought if animals or humans get exposed early in life, for example, then there is no allergy development here. So it's a way out very early, there will be even no sensitization. Or for the animals that get sensitized here, right? So the ones that have IgE on their mast cell surfaces are sensitized, they can also develop similar mechanism, really downregulates their sensitization, and again, no clinical allergy. So there are several ways and times of influence here if you think about allergy. So it's a very complex disease. This is our working model, as I said. And so the next thing that we did and how we started this project is that we looked at neonatal immunity. So we realized that neonatal immunity is quite different than the immune response of an adult. And so it's very complicated, it's probably not as well investigated. It's the same for humans as for animals. no big differences there. And one typical thing that we see if we look at the immunity in a very young foal, for example, and these are horses, so foals, different times of age. So taken right after birth, and then 5 days, 6 weeks to 12 weeks of age, and then adult horses. So we took their cells, their white blood cells. This is what this picture shows. It's a fax picture, and then the white blood cells were stimulated, in this case with PMA, which is a stimulus that's usually used to provoke T-cell responses in animals or humans, in cells. And so we looked for key cytokines of the T-cell response. So one is interferon gamma, which is a typical cytokine of the Th1 response, and the other one is IL-4, which is a typical cytokine of a Th2 response. So the Th2 response is the driver of allergic responses, while the interferon gamma response is a driver out of an allergic response or a driver of antiviral responses. So quite different arms of the immune system. So if you look at the adult horse here, if you do this polyclonal stimulation, you see that the cells are able to respond with a lot of both cytokines. So a lot of interferon gamma is produced, and also quite a bit of IL-4 is produced. So perfectly capable to do those immune mechanisms. If we look at the neonatal foal, so shortly after birth, they don't respond. So they have T cells, they have CD8 and CD4 T cells, that's all fine phenotypically, but the cells don't respond to those common stimuli in the same way as the adult horses do. There's no IL-4 at all, and just a very tiny amount of interferon gamma. And as time goes on, so that increases a little bit for interferon gamma especially, but not so much for IL-4. So if we looked at that, that's just one individual. If we look at that in many horses, it looks like this. So for interferon gamma, we see a slight increase, and then it hangs in there. And there's still at 3 months a difference to the adult a little bit, but over time of 1 year, that adjusts. So for IL-4, it looks actually quite different. So that stays the same almost all the time. It's still remarkably lower at 3 months of age than in an adult horse. There's a little peak at 5 days. So we were wondering what that is, and if this missing Th2 response is actually responsible for the missing IgE in the very young foal. And the answer is probably yes. However, so we looked at those IL-4 producing cells at day 5 a little closer. So this is again a fax picture that shows IL-4 here on the y-axis in this case, this little population. And then we phenotype those cells to find out what are these cells. Are those really T cells, or is this something else? And so T cell markers are CD4 and CD8. You can see that these are not positive for IL-4. The positive population for IL-4 is a non-T cell population, basically. So it's non-T cell, it's not IgM positive, so no B cells. It has no MHC class 2 on its surface, but it is positive for IgE. So those are IgE positive cells that produce IL-4 very early in life of the foals, and during a time when the Th2 production is basically not there. And those are perfectly healthy foals, so they're not sick or anything, it's just their normal immune response that they have. So we try to identify those cells a little better here. What are these IgE positive, IL-4 positive cells? And so we did an MHC sort in that case, because it showed that those cells are also negative for MHC class 2. So we took the MHC class 2 negative population here, and then sorted it by IgE. These are the IgE negative, and these are the IgE positive cells. So that's the population we were looking for. And then we looked at them in a very simple diff spin, and those are all basophils. So the IgE positive IL-4 producing cells after birth are basophils. Basophils are cells that have been, oh no, I'm too fast. So the basophils actually, so this shows the same thing again, puts the cells in cell culture, and then looking at the supernatants of the cells after stimulation with or without IgE. So if we take those fractions apart and we look at the IgE positive MHC class 2 negative population, which contains the basophils, this is where the IL-4 comes from. While the other fractions, like the MHC positive cells where the T cells are in, they are not producing any IL-4 in foals. So absolutely no production by T cells at that point. This is too far, right?
It's too far.
Yeah. So the basophils, those are specialized cells. They are innate immune cells. They are usually cells that are associated with spitting out inflammatory mediators, causing inflammation, and things like that. And they are very similar in that respect to mast cells. So this is another cell type where sensitization can occur. They are involved in antiparasite responses and in allergy. And more recently, they have been involved in a role in immunity, basically, and they have been thought to be the cell type that actually induces the initial Th2 response. So for our horses, we could not support that because we do see those cells at day 5, but then we don't see a Th2 response for several months. So in foals, they have another role. They do something differently. They are not just Th2 mediators. So this is showing the experiments in foals here in that case. So that takes foal and mare cells, and they were stimulated with anti-IgE. And if we look at the histamine release in those cells, we do see that basophils in foals do respond quite differently. So they don't do this histamine, this inflammatory mediator release. So it's not the classical inflammation driving cell type, while the mare cells do that. So they release lots and lots and lots of histamine in that case. However, if we look at IL-4 in both populations here, so that's pretty similar. So the foal cells do a lot of IL-4, and similarly the male cells do. So which means the foal basophil is kind of driven towards immunoregulation and not so much towards inflammation. So that gives that cell type a quite different role in the foal immune system, and to be very honest, we still don't know what the role is. It's not Th2 driving, but it's certainly IL-4 production early in life. So in addition to that, when we looked at those cells, in that case at the MHC class 2 positive cells, we found that the foal cells make a huge amount of another cytokine, and that's IL-10. So IL-10 is a regulatory cytokine, one of those immunoregulators that has been put in context with protection from allergy and downregulation of allergy and basically tolerance. And that's what these cells can do. So the foal cells, in response to anti-IgE, which is a very similar stimulator as an environmental antigen in this case, can produce a lot of IL-4, but they can produce even more IL-10, and they don't react towards inflammation. So quite a different cell phenotype. So with this information, I came here in 2009. And I talked to Sister and Simon Willi, and I explained this to them, and I still know that they looked at me and found this very interesting, and then we started to think about this project, what we can do and how we can solve this and how we can find out about these different cell types and how they influence allergy. And there's one more thing to it, and that's this. So I told you already, foals don't make IgE until they are 6 to 12 months of age. So there's no IgE in foals. So how does it all come that I stimulate a basophil with anti-IgE and they respond? So that is, the reason for that is that the mare has a lot of IgE in the colostrum, and they transfer this with the colostrum to the foal. So the foal gets this IgE, and within a few hours after they take up colostrum, we can detect that IgE in the circulation of the foal, so in serum, and we can also detect it on the cells of the foals, which means on the basophils. So the IgE on the basophils in a neonate is maternal IgE. It's not foal IgE. So it transfers, what the mare does, it transfers its own adaptive immune response, so the accumulated immune response that the mare has over her whole lifespan to the foal at that time. And that's on the surface of the basophils. So if there's cholicoides in the environment of the mare has IgE to cholicoides and is sensitized herself, she will give that to the foal. So the foal cell can be passively immunized, basically, or sensitized by this maternal IgE. So we were thinking that this may play a role, actually, in the development of allergy. And if you look in the human literature in the developed countries, like the USA, they say no maternal IgE from allergic mothers, so that's a risk factor for children to develop allergy. So we're not entirely sure about that, if that's true. Also, there are epidemiological studies that support that. So we developed a model that is based on the maternal IgE transfer here, and it has two arms. So it has a basophil on one side, what we observed. There's a maternal IgE on the surface, and if this is cross-linked by an allergen or by anti-IgE, we see IL-4 production. And at that point we thought it's for Th2 development, and maybe that's true or maybe not. And the other cell that's involved here is a monocyte in that case that also binds maternal IgE, and that is the cell line actually that, or the cell type that produces this high amounts of IL-10 that I just showed you. So, and that is an inducer of Treg cells. So those key players that really are important to decide or to make the decision for the immune system if allergy is developed or not developed. So the question was, is maternal IgE something that is supporting allergy in a foal by driving Th2 responses? Or is this rather something that also protects the foal by driving very early on allergen-specific IL-10 responses in those animals? So is this actually something that's needed for the animals to develop less allergy by making this immune recognition early and driving it into a regulatory tolerance rule rather than overreacting? Since these basophils at that time in the foals are not cells that will spit out a lot of inflammatory mediators, so they are not driving allergy. So supporting to that was also the observation. This is a general prevalence slide. I would like to point out this here. And was supported by the observation that if horses are imported from Iceland as adults, young adults, they develop a very high susceptibility to disease. So you will find many horses that develop disease. Some countries reported 60 to 70%. While the horses, when they are bred in Europe, they have much less disease. So by now that is supported by at least 3 or 4 epidemiological studies that show exactly these data. And this is one other mare from us that's allergic. So we want to know if the maternal IgE, so the non-genetic factors, and the acquired immunity of the mother, which is given with the IgE to the foal, or purely the time of exposure to allergy is giving us this difference. If you look at those numbers, you could of course argue that it's good to have maternal immunity, because these horses that are born on the continents, they are born to mares that already have immunity to culicoides. They have been exposed, they have immunity, so the maternal immunity is transferred to the foals and can act through this mechanism to influence that disease. So early allergen exposure to maternal immunity. So we developed a model with Iceland and the US here, Cornell, where we looked at first culicoides exposure as young adults, high risk. So this is basically what the epidemiological data tell us. Foals are born without maternal immunity to culicoides, compared to early in life exposure, low allergy risk, and the foals are born to mares with immunity to culicoides. And then there was one group missing to figure that out. So we thought about if they get exposed early in life to culicoides, but they have no immunity to culicoides, so what would they do? So this was a question to answer actually, is early life exposure or is maternal immunity an important factor to decrease allergy risk? So we don't know about this group yet, because they are not old enough, but those are our 3 experimental groups. And this is how the project that's called the Big Mare Project, the huge mare project here. So it's called the Icelandic Horse Project at Cornell. And this is what it is actually. It's a uniquely controlled culicoides exposure and allergy model in horses. And it's a long-term study, as I've pointed out several times already. So we did a huge effort by bringing horses from Iceland to Cornell and putting them in these 2 groups with different exposures. The horses for this group actually were horses that were transported to the US in the mare, so in utero basically, and were born in the US in the first summer when the mares were there, so before the mares developed immunity. And so that's how this unusual group was created that we would usually not see in the real world. So the experimental study is that we have a full sibling study. So all the offspring are from one stallion. That's a stallion. Geisli, and I stop here. I think I cannot say that. And and from 50 non-related mares, and there are 3 full sibling foaling years. We only lost one of the offspring, so we have 3 groups of 14 full sibling animals. The first group got no maternal IgE, and they were not exposed to culicoides, so were born here at Hvernig in 2011. They were first exposed at 2 years of age when they came to Cornell in 2013. The second group was born at Cornell. They traveled to the US in utero. They got no maternal culicoides specific IgE, but they were exposed directly that summer, so early environmental exposure for that group. And then the third group mimics the group that's born on the continents in general. So they got maternal IgE, the mares were there for a year already, they were sensitized to culicoides and developed immunity, and they also were exposed to culicoides at birth. So the unique thing about the study is that this is that these horses have a very close genetic relationship. So it's closer than you would see in the normal horse population. It's closer than you would also see in the human population, but it's not as close as you see in an inbred mouse. So from the model perspective, it's heterogenetic enough to mimic a human model, but it is still kind of controlled, because we know allergy is affected by many genetic factors, and so we don't want to have too much variation in the genetic factors, because then it gets too complicated with a relatively small n to analyze that data. So, and then all the animals are only kept in one, or were kept since today, in only one environment with culicoides exposure. So the exposure is very much defined for these animals. They're not at different places with different times of first exposure, etc. So always the same. So in 2009, we talked about that here at Caldo, actually developed the project. 2010, the mares were bred first. At 2011, and here are the pictures from 2011 when the foals were born here, and some of you may remember that. Actually, here blood samples were taken and shipped to Cornell, except for some time when events like this happened, which we have all the time here, obviously. So happy mares and foals here at Caldo. And then 2012, early in 2012, after fighting with some snow on the roads, and finally it all worked out, that the mares were loaded on transport containers at Keflavik, and they traveled to the US, came to Cornell in this one big transporter. We made a specific and unique arrangement with the USDA that's here on this little sheet. This is an import permit for horses to the US to bring the horses directly to Cornell, and to avoid that the horses would go to the official quarantine facilities. So they made it directly from the plane into the transporter to our barn, which is entirely isolated. So these horses that are in our facilities still haven't seen a single horse in the US until today. So we use this to develop also this unique infectious disease model in the horses. So this is Geisli being unloaded at Cornell. They traveled under APHIS seal to make this all possible. And this is a facility at Cornell where they are kept, so very isolated. It's the airport is located here. There are no houses around here. There are no other horses around here. A little bit of wildlife, of course. They go in and out. We use a lot of care going to these horses. We never go there from another horse barn. We always shower in between and change, so to keep the horses infectious disease free. And so they have 30 acres of pasture here, a little culicoides breeding area. That's a pond, right? This is where we get our natural exposure. And so when we have these barns here, the 2 barns with the blue ceilings, those are the places where we sample the horses to bring them in. Otherwise they live outside, like they do in Iceland, 24/7 and all year round. They also gave birth outside, so this is from the foaling 2012 at Cornell. A couple of our foals just born at night. So we had foal watch all the time to get them early, to get the colostrum samples and the pre-suckle samples from the foals. We had a lot of fun with my collaborator at Cornell, large animal internist, Dr. Perkins. So waiting a foal, right? So it was very fun. Summer, many students involved in sampling and helping with the horses during that time. So during all that time, the horses are analyze. We take many samples over the time. We do a lot of experiments to figure out all these questions that I just showed you. We don't have the answers yet. The project goes till 2016. So the horses need to develop their phenotypes. We judge them at 3.5 years of age. And then basically you can take your data apart and analyze them in 2 groups. So it's really a long-term project. However, during the whole time at Cornell, they are scored. So we determine their clinical allergy score and status all the time. And that's how it's done. So it looks at if they are itchiness, pruritus, it looks at hair loss here, alopecia, and at skin irritations and dermatitis, and they end up with a total score. And that's happening several times a month to give us an idea which horses are clinically allergic and which are not. And there are photographs and everything is taken to document that. So to give you a few pictures here, so many horses itch, for example. So that's not an allergy. This is one of our yearlings born in 2012 in a 2nd year of exposure, and she rubbed her tail a little bit, and she also has this piece here at the mane, but really no skin irritations. So this is not an allergy. And so looking at these studies that have been reported, if owners judge this, sometimes I have heard people that say, oh, this horse itches its tail or scratches its tail, so that's allergic. That's of course not an allergic horse, right? There belongs a little more to allergy than this. So this is why we do the scoring on them all the time. These are the 2-year-old horses that came in 2013, and you can, this is not the best picture, but you can see our different insect species here. This is just a Culex midge, I would say, mosquito, housefly, and this is Culicoides, here this tiny thing with a red belly, so it's sucking blood already, a little difficult to see. And that's what